Hi, my name is Olivia Piacentini, and I'm going to be going through all the stages of mitosis and cytokinesis. During interphase, the phases G1, S, and G2 occur. During G1, also known as gap 1 of mitosis, the cell is growing, metabolizing, and possibly differentiating. Although the cell is metabolically active and growing, no DNA replication is taking place during this phase. The S phase of mitosis, which is also known as the synthesis phase, is where replication of DNA occurs. A complete copy of DNA is synthesized in the nucleus. Centrosomes are also replicated within the S phase. During G2, also known as gap 2, the cell can continue to grow, metabolize, and differentiate. Also, during G2, the cell is checking the synthesized DNA and making any necessary repairs to the double-stranded structure. During prophase, the centrioles will divide and move to begin to establish poles in the cell. The initial formation of the mitotic spindle will occur as well. The nuclear envelope will begin to break down as the broken down contents are released into the cytoplasm. Chromosomes condense and become visible. Sister chromatids are the two parts of each chromosome. They are connected at the centromere and held together by a multi-subunit protein called cohesin. In prometaphase, chromosomes will move to the equatorial plate, or metaphase plate. The centrioles will reach the opposite poles in the cell. Mitotic spindle is formed by shooting out microtubules that connect to the centromere of a chromosome. Fragments of the nuclear membrane are visible. During metaphase, centromeres are aligned on the equatorial plate halfway between centrioles. The spindle fibers bind to the connecticore as the chromosomes migrate. Connecticore microtubules have one end near the chromosome region and the other end anchored to the connecticore. The centrioles are at opposite poles. During anaphase, disjunction occurs. Disjunction is when homologous chromosomes move apart, the centromeres will split, to the opposite pole where the centrioles are located. They move through the shortening of spindle fibers. The separated sister chromatids are now called daughter chromosomes. In telophase, the nuclear membrane will reform where each daughter cell contains identical chromosomes to the starting parent cell. Centrioles will disappear during this phase. This phase signifies the end of mitosis as the nucleus has divided. Cytokinesis occurs as the cytoplasm that was previously contained in a single cell divides into two daughter cells. Each daughter cell contains identical genetic information in the nuclear membrane as the parent cell. In animal cells, this is called cell furrow because a furrow in the plasma membrane is formed. The main difference in cytokinesis in animal and plant cells is that plant cells will construct an additional structure called a cell plate. In plant cells, a cell plate is created between the two daughter nuclei as mitosis is completed.